I would be remiss to tell you that I'm only a director of research. I live and breathe science. And my research actually is just, for your purposes today, an introduction of some of the areas that we're interested in working on as well. I have actually 4 main interests in my laboratory, and I just want to briefly introduce you to the different areas of research that I'm going to continue here at the Masonic Medical Research Institute. Primarily, my research actually is a signal transduction laboratory, but what that means, and was partially introduced previously by the other colleagues, is that this is the mechanism by which something from the outside of a cell actually transfers and moves inside that cell to go into a nucleus and to tell that cell exactly what it should be doing, whether it's continuing to grow, to kill itself, to move to a different location, or to activate other genes that would be responsible for other purposes in perhaps even other cells. And my research has primarily focused largely on rare disease and pediatric disease. Pediatric RASopathy disorders is what we call them, because they primarily affect this canonical signaling pathway called the RAS-MAP kinase signaling cascade. It's actually quite an instrumental pathway because it is responsible for virtually all cell proliferation and viability in almost every single cell type in the body, including cancer cells. And so therefore, I can manipulate the kind of signaling in the research that I do from one cell system to another actually relatively easily. But by studying rare disease, it actually gives us an opportunity to really understand what manipulation of this particular kind of pathway can have and what deleterious effects it could produce in human tissue. And so these are rare diseases. They include things like Noonan syndrome, Leopard syndrome, or Noonan syndrome with multiple antigenes, and other associated RASopathies, including Costello, neurofibromatosis, cardiocutaneous syndrome. They're all along the same family, and while uniquely individually rare, collectively they're the most common form of disease and cardiovascular disease in live births today for human patients. So this is an example of a particular kind of patient that we're interested in. They have very characteristic facial features, craniofacial abnormalities, low set posterior ears, ocular hypertelorism, a web neck sometimes. They have short stature. Wow, there's the light. Yeah, see? The time's up. Short stature, and they also have very significant cardiovascular defects, which is how I ended up studying these patients. My background is actually in cancer. So these children are also unfortunate enough to also develop various types of leukemias. And my original research in my postdoctoral fellowship primarily focused on understanding how they were developing these cancers. And I noticed as we were studying them that they were very uniquely different in their cardiovascular manifestations. And so I transferred my work into a cardiovascular space. But we're very interested in using these diseases, as I mentioned, to look at other aspects. We primarily look at this in a tissue culture manner. We have generated 22 different mouse models that recapitulate the various rare diseases that I've described to you today. And we're using those to reproduce the phenotype that we see in human patients. We've also recently adapted a new model system that was introduced to you by both Gary and John, which is the Inducible Pluripotent Stem Cell System, which is, I mentioned earlier on some of the tours, a very unique way that we can actually assess and study human disease in a human context. So you basically can take any skin cell, epithelial cell, which is a saliva or drop of blood, and using what's called Yamanaka factors, Yamanaka being someone who won the Nobel Prize for being able to de-differentiate a cell from a known purpose, like a skin cell, into a nascent cell that can become anything with the right conditions, is what we call inducible pluripotent stem cells. Then using a variety of growth, using a variety of growth factors, we then can manipulate these nascent cells to turn into anything we're interested in. Gary talked about turning these into cardiomyocytes. Our laboratory is interested in also studying other aspects of disease. So right now we're differentiating these into T cells, gastrointestinal organoid structures, and also cardiac fibroblast and cardiac endothelial and epithelial cells, so we can understand specifically what the role of these particular cell types are in the development of a particular disease. The other asset in studying these, Jason mentioned a little bit earlier, is being able to use these cells to then use either global, large-scale, or small-scale therapeutics to assess whether or not these could be applicable drugs that we could use in human disease. This is exactly where the field is moving. It's precision medicine. We can actually look at someone's specific disease etiology in the background of their genetic code, because sometimes there are various genes that actually affect the disease. It's not that one genetic mutation, it's sometimes the epigenetic or the variants in other genes that actually contribute to that disease phenotype. And we're in the process of studying and understanding that further as we go along. I mentioned we're also interested in gastrointestinal disease, and this is how we're going to do it, is through this IPS system. We again go back into the mouse model, which is critical for validation in a mammalian system, because after all, this is still cell culture, and as Gary and John mentioned, some of these cells don't mature to the proper properties that you would normally see in an adult phenotype. The organoid is not in that way, and using this system where you can then take these inducible pluripotent stem cells and really generate what is in a dish, your gut, is an instrumental way to study diseases like Crohn's disease, which is an autoimmune disorder. We're also studying gastric cancer in this capacity, and we're also looking at things like what causes diverticulitis. In this context, these rare patients have been instrumental in guiding us to this point, because it turns out that whether you activate that pathway I talked about or you have a mutation that turns it off, dictates whether or not you get a gastric cancer or you have a diarrhea type of phenotype like Crohn's disease. So it's a very important way for us to actually parse out the mechanisms that cause these other more common disorders by looking at these various rare disease patients. The next project I want to talk about is something Jason mentioned, is a collaborative project that we started after we had noticed a particular phenotype in a model that we had made on a protein called RhoA. It turns out that this enzyme is actually critical in the transition from the compensatory, where the heart tries to make up for an additional stress, such as myocardial infarction or pressure overload, to transition it into something called heart failure, which is the point at which you need a heart transplant or you start to get edema in the legs, and is a clear indication that you need some intervention, surgical intervention to prevent death. It turns out that this enzyme was really interesting because although it caused by echocardiograph, which is the machine that Jason mentioned we'll be getting onto the third floor, shows a very significant increase in the spacing here. This is how, so when you think about an echo, this is a normal echocardiograph. The various contractions here actually make it look like an ovular shape. When you have a cardiac dysfunction, such as cardiac hypertrophy, where the walls get thicker, what happens is the contraction is still continuing because it's compensating, but the space within the ventricle for the blood to pump actually becomes reduced because the wall around it is actually much thicker. In heart failure, the heart, you can think of it more like a rubber band. If you continue to stretch that rubber band over time, it loses its elasticity, and you end up from something that was a small, round, elastic rubber band to something that's now really stretched and unable to contract. And that's exactly how you think of the heart, because in heart failure, you're no longer contracting, and the blood ends up getting pooled into the ventricle, and that's how you get edema, you get some circulation concerns, and other reasons why it's a critical problem in the medical field. So despite the fact that this particular enzyme actually caused this heart failure phenotype, the interesting thing about it is this was the first evidence that we could separate heart failure from something called cardiac fibrosis. And you heard a little bit about this from my colleagues today. Cardiac fibrosis is actually the part of the tissue that actually dies in response to a particular disease etiology. So even though we had heart failure, there was no fibrosis in this particular model. So it allowed us to divide up and study cardiac fibrosis specifically as a potential therapeutic, because usually fibrosis leads to heart failure. And so my collaboration with Jason on this project is using his nanoparticle technology to isolate drugs that treat fibrosis through the molecular mechanism, the signaling pathway we define to cause this problem, and target it specifically to those cells, so we can use it as a therapeutic. And lastly, this is just my last slide, I also work on autoimmune disease. This was also very serendipitous, again from the rare disease patient cohorts that I actually have been studying for over 20 years. Many of you in the room already know my mother passed away from systemic lupus erythematosus about 9 years ago now. And before she died, she had suffered from this disease about 26 years. And many years, always asking me, you're studying cancer, you're studying muscles, you're studying heart, when are you going to do lupus? And I said, I just don't see a connection. The year she died, about 2 papers were published that talked about how the patients I had been studying for 15 years were also getting lupus. So the same kids who are unfortunate enough to get cancer sometimes also sometimes got this devastating autoimmune disease. And so I said, I need to look at this. At the time, down the hall from me happened to be one of the world's experts in systemic lupus erythematosus. He will also be one of my consultants here when I create my scientific advisory board so that we can continue these collaborative efforts. Provided me with some animal models to look at and study lupus. Principally, this disease occurs in women of childbearing age. It causes deleterious effects in almost every single tissue, including the heart, which is why we became initially interested, although we've become systemically interested in this disease. And it turns out that the reasons why women are affected, and primarily women of color and women of Asian descent, is unclear and also an area of keen interest because it may be hormonally based. But we discovered that the mechanisms that cause those rare diseases were also the same mechanisms that cause lupus. And we're currently in discussions with GlaxoSmithKline on development of a drug therapeutic to treat lupus syndrome patients, because I realized I took the slide out. I could show you the phenotype of the animals that were treated with the drug that we developed with our collaborators at the University of Indiana, completely ameliorating the pathogenicity of this disease, which is remarkable since this disorder has no treatments to date. So those are just some of the areas of research that you can see from my new group of fantastic scientists, along with the people who are now going to be coming forward, are going to be revolutionizing the Masonic Medical Research Institute in ways that had never been imagined before. And I appreciate your support and your constant